You're listening to Conferences Online Allergy from Children's Mercy Hospital in Kansas City, Missouri. Today is December 23rd, 2024. Our topic today is Abbas Chapter 10, Differentiation and Function of CD4 Effector Cells. This is presented by Dr. Nikita Raje. She is an Associate Professor of Internal Medicine Pediatrics and is the Chief in the Section of Allergy Immunology at Children's Mercy Hospital in Kansas City, Missouri.
November 2024. I'm Nikita Raje from Children's Mercy Kansas City and UMKC, and we'll be talking about chapters 10 and 11 from Abbas today. First chapter, chapter 10, is on differentiation and function of CD4 effector T cells. Definitely getting into the chapters that are more relatable, I feel like, after some of those difficult chapters that we went through. But stop me anytime if if you want me to repeat the information or if some of the terms don't make sense. Here are the disclosures, nothing relevant to the talk today. And then learning objectives is to expand our understanding on overview of CD4 T cell mediated immune responses. We'll talk about the subsets of CD4 cells and then functions of other T cell subsets. Few questions today that we'll go over at the end. Major function of Th1 cells is to activate macrophages. Which molecules expressed by Th1 cells are essential for this function? Which of the following is one way a cytokine made by Th2 cells contributes to atopic disease? I feel like there was one more question. I don't know what I did here, but anyways, we'll come back and maybe we'll have 3 questions at the end. So let's talk about the function of CD4 effector T cells. So remember, just kind of to recapitulate, we talked about naive CD4 cells, naive T cells, single positive cells, and their development in the thymus. They get released into the circulation when they are recent thymic immigrants, and then they become regular naive CD4 T cells. They still haven't met their antigen. When they recirculate through lymph nodes and other secondary lymphoid organs, they may come across the cognate antigen that's specific for them. When they do that, they get activated. When they come across that antigen, they interact using all the surface molecules, including TCR and the CD3 molecules and zeta chains that help in signaling, as well as the second signal that they use. All of that helps in an interaction in that immunological synapse. That activation leads to proliferation of those cells and then differentiate those cells into the effector T cells. So now we know that these effector T cells are formed, either the effector T cells that are the Th subsets, then they can also become a memory T cell. And so we are going to focus today on the function of these effector CD4 T cells. So the function of these cells is to help with certain specific functions of cells that they interact with. So for example, for phagocytes such as macrophages and neutrophils, the CD4 effector T cells can help recruit those cells and then activate those cells. Macrophages important for intracellular microbes, neutrophils important for extracellular microbes. And then other leukocytes as well. They help in recruiting them and taking them to the sites where they're needed, whether it's eosinophils or monocytes or basophils. So that's the function for all these white cells. Apart from that, they also help other lymphocytes. So they help B cells in making high affinity antibodies and then help them in class switching. And they help the CD8 cytotoxic T cells in their function of cytotoxicity or killing the infected cell or damaged cell. So here the figure is just showing how that interaction occurs. CD4 T cells interacting with macrophages. Now remember, macrophages are one type of antigen presenting cell. So macrophage presents the antigen, CD4 T cells recognize that, they get activated, they get that second signal, and then once activated, they actually help in activation of the macrophages themselves. So they license them further to activate the other CD4 cells, and they also help macrophages get more activated so they can help in their phagocytic function. Apart from that, here there is that interaction between the CD8 T cells with their target cell, but that interaction is also helped by the CD4 cells. So I believe I talked a little bit about this process where the effector CD4s go. So we talked about this last time, I think. So remember, I talked about how that activation occurs in that secondary lymphoid organ. So when that activation of naive T cells occurs is when they proliferate and then differentiate into effector T cells. So these effector T cells are generated in secondary lymphoid organs. And then they migrate to peripheral tissues where they are needed. They do that. The migration occurs because of presence of adhesion molecules and chemokines. So again, remember, that's like the GPS that kind of guides these cells in where they are going. Once they reach the tissue, they can recognize the antigen there, and they have to be retained in that area for a longer time. So they have to have affinity for some of the adhesion molecules in that tissue, such as VLA might help interacting with fibronectin, or CD44 with hyaluronan, and it helps in retaining them in that space. So T cells, effector T cells, perform their function in that peripheral tissue. Once they have done that, they are gonna, continue to help with that function, but the cells with other specificity are continuing to recirculate, and then those might die depending on how long they have recirculated and if they have not come across their cognate antigen. Versus the cells that are in the periphery, effector T cells, they may perform their function and they may die once that function is over. Some of them might actually transform into being memory cells as well. Now, some of the activated CD4 cells, they do not go to the tissues, but instead stay in the secondary lymphoid organs, and they move towards the lymphoid follicles. Remember, when we talk about follicles, we are talking about B cells. B cells are the ones that make the follicles, and that's their workshop, basically, where the B cells make antibodies. So the T cells, activated CD4 T cells that move towards the follicles, some of those will help the B cells in their function. These are called follicular helper T cells. So this is just an experiment that shows how this function of CD4 cells was explained. So it's cell-mediated immune response against phagocytosed microbes, that is with macrophages, interaction with macrophages, how that comes into effect. So that was explained by this experiment where they looked at infection with listeria, and they saw if the immunity against listeria can be transmitted or transferred from one person to another using T cells or just the serum, so non-cellular part, right? Which means we are talking about antibodies, and can that infection be cleared? So it was seen that when T lymphocytes, so looking at this figure here, when T lymphocytes were transferred, the viable listeria went down. So non-immune T cells did not make a difference, that's the blue line, but when immune cells were transferred, there was specific immunity against the listeria that was helpful to keep the number of viable listeria down. When serum was used, there was no change because serum does not help with clearance of listeria. Listeria needs cell-mediated immunity. And then the third figure here shows the difference between how the killing occurs if there are just the immune cells versus immune cells along with activated macrophages. So you can see how if the blue line here, light blue line here is activated macrophages, the number of so this is not the same thing. It's not the number of viable listeria. It's the percentage of killing of listeria and how that's higher when there are activated macrophages instead of just the immune T cells. All right, so we know that this function of CD4 T cells in helping the phagocytes occurs because of that signal, second signal, CD40 ligand with CD40, along with some of the cytokines that are released. This interaction helps with delayed type hypersensitivity, and that's because of the action of CD4 cells, then recruiting leukocytes, and then that actually leads to damage of some of the normal tissue that you can see in TB test or similar experiments with Candida antigen. All right, so now we come to the important aspects of the effector CD4 cells, where depending on the type of activation and other factors, there are diverse immunological reactions that occur that because of the function of CD4 cells. So that's provided by these subpopulations of CD4 cells, and they are numbered. So T helper subsets are numbered Th1, Th2, Th17, follicular helper cells, Tregs. So all of these have specific markers and specific functions and interactions. So Th1 subset releases interferon gamma. Now, I think that one more way of remembering this is, what does interferon gamma do? It helps with macrophages, and Th1 cell, the principal target cell, which the Th1 cells help, are macrophages. Th2 cells release IL-4, 5, and 13, and they take help from these cells, eosinophils, to provide Th2 response. Th17, the important cytokines for this pathway are IL-17 and IL-22, and they are important for recruiting and function of neutrophils. T follicular helper T cells, cytokines are IL-21. Some of that function is also helped by interferon gamma and IL-4, and they help B cells. The target cell is B cells that they help. And based on the cells and the cytokines, you know that the immune reaction will be specific to that particular cell. And I think the most important information to remember is this column here. You want to remember, and we'll talk about this more in detail with each pathway, is what each cell, T helper cell, is targeting. So Th1 cell is targeting intracellular pathogens, Th2 helminths, Th17 extracellular pathogens such as bacteria, extracellular bacteria, and fungi, and then B cells as well, extracellular pathogens. Of course, some of these cells can cause damage, and that can Th1 cells in that case will cause chronic inflammation or autoimmunity. Th2 can cause allergies. Th17 again autoimmunity and inflammation, and then autoantibodies from B cells can lead to autoimmunity as well. So some of the features of these Th subsets, some of the common themes are that Th subsets produce combination of cytokines And some of the subsets may produce cytokines that are not characteristic for any of the subsets, such as IL-9. Maybe we can call them TH0. And some of these subsets have plasticity, which means they can move from one subset to another. Same cytokines can also be produced by the innate lymphoid cells or gamma delta T cells, which means these cytokines are not specific just to the Th subsets. And then again, for where these cells end up, that depends on their function. Where are they needed? So the homing patterns, remember we talked about this concept, the cells home to a tissue based on what kind of chemokines and chemokine receptors are present. So Th1 cells express CXCR3 and CCR5 on them. That leads to them homing to some of the tissues, and they also use E and P selectin ligands. Th2 subsets can express CCR3, CCR4, CCR8, which helps them to home to the mucosal layers. And then Th17 can express CCR6, the CCL20 that's produced by tissue cells and macrophages get attracted to, the cells get attracted to those areas with CCL20 because they express CCR6 on them. All right, I don't expect you guys to know all these particular chemokine receptors and things for each subset, but you get the idea that where are they going to end up is based on those chemokine receptors, but the bottom line is Th2 cells will end up in the mucosal layers versus Th1, Th17 might be in specific areas as well. So how do these CD4 T cells, when they become effector cells, know which type of Th subset to become? And that process of differentiation of CD4 effector cells or CD4 cells once activated to become these Th subsets has a specific type of process. The steps of that process include induction, then commitment, and then amplification. So 3 steps. So induction means that there are certain stimulation that these cells receive that will lead the cells to become particular type of subsets. So for example, antigen presenting cells, NK cells, mast cells, all these innate cells produce cytokines based on the type of microbe that's present at that time. Those cytokines are going to act on the activated CD4 cells, and that induces some of the transcription. Remember, we talked about the signaling. Cytokines act on the JAK-STAT pathway, and that actually causes signaling and transcription of certain genes. Those transcription factors are the ones that are going to be activated, go to the nucleus, act on certain genes and promoter regions of certain genes that will make certain proteins. Also, there is some genetic makeup that leads to pulling of the cells into either Th1 or Th2 direction. But these cytokines leading to the signaling, leading to the transcription of certain genes, leads to the CD4 activated T cells to become a particular subset. That's about that background of genetic makeup. Once they are induced to become Th1 cells or Th2 cells, they have to commit. There are transcription factors in those cells that are going to be important to decide, to kind of make sure that they are committed to become one type of cell or the other. And the epigenetic modification of some of those genes help fix that whole state into an active state transcriptionally so that they are committed to become that particular subset. All right, and then the genes encoding some of the cytokines will keep the genes needed for other subsets to be inaccessible. And then the third step is amplification, which means the cytokines that help in promoting the cells to become one subset will inhibit the differentiation towards other subset, which means, say there are Th1 type of cells, releasing interferon gamma. Interferon gamma will help in that positive feedback and commitment to become Th1 cells, but they'll also inhibit the cells around them to become Th2 or Th17. IL-4 from Th2 subset will inhibit Th1 differentiation, and IL-21 promotes Th17 and inhibit Th1 or Th2 subset differentiation. Questions? I stop here because now we are going to move into talking about subsets, and we're going to build on this whole concept of differentiation and how these cells differentiate and function for particular microbes. Okay. All right, moving along. Stop me if you need to talk about anything or need me to talk about, repeat some of the information. So let's talk about the Th1 subset. Like signaling, I also want to kind of use the same concept of thinking about the last step first. So Th1 subset, sure, they come from the activated CD4 cells, but I want to keep in mind what's the motivation for these cells. The motivation comes from the microbe. If there is a fungal infection or extracellular bacterial infection, can someone tell me what subset is needed to fight those microbes? We are just talking about 3 subsets, so you can take a guess, but we are talking about extracellular microbes or fungal infections. What subset is needed to fight those microbes?
TH17.
TH17. If it's a helminth, what is the subset needed?
TH2.
Th2.
And then if it's intracellular microbe, what subset's needed?
Th1.
Awesome. All right. So that motivation for the cells to become a particular type of subset depends on the microbe that's present. So here we are going to talk about intracellular microbes, microbes that are ingested and then they activate the phagocytes. So if that kind of microbe is present, say tuberculosis or listeria, leishmania, viral infections, all of these are intracellular infections. In presence of those microbes, there is motivation for T cells to differentiate to become Th1 subset. So in presence of some of these microbes, some of the cells around them, like phagocytes, dendritic cells, all the antigen presenting cells, will release certain type of cytokines such as IL-12 being one of the important one, and then IL-18, type 1 interferons, particularly for viruses. So these cytokines are gonna make an environment, right? There is a cytokine milieu in that presence of that microbe. So here, looking at this figure here, say there is a intracellular infection, that microbe is going to be taken up by these antigen presenting cells, go to the lymphatic vessels and through the lymphatic vessels into the secondary lymphoid organs, where they'll come across this naive T cell. When they come across, during that time, they're going to release those cytokines, right? Dendritic cells, macrophages, they're going to release IL-12 because that's what happens with intracellular microbes. IL-12 is going to act on these cells, and in presence of that cytokine, in presence of some of the second signals that these APCs provide to these naive T cells, telling them that it's an intracellular infection and what kind of infection, these cells will release interferon gamma. The interferon gamma can also come from NK cells, and that is going to act on the differentiating CD4 cell, and that signaling will lead to some of the transcription factors that will get activated, including TBET, STAT1, and STAT4. Because of that TBET, that's going to help in transcription of particular genes that are important for Th1 subset, such as interferon gamma. It will also help in IL-12, which will provide that positive feedback. Th1 subset releasing that interferon gamma will help in the amplification path. Remember, there was that induction and then commitment, and then there is amplification. That amplification occurs because of this cytokine interferon gamma. That's the most important cytokine that's released from Th1 subset. That's going to help in interacting with the interferon gamma receptors on the cell itself. So that's your positive feedback, but also it will help. Interferon gamma is needed for the function of macrophages, so phagocytes. What do phagocytes do? Phagocytes are going to be important to kill the microbes that are intracellular, right? So we came back a full circle here. We started with the microbes releasing interacting with APCs that will release cytokines that are going to motivate the activated CD4 cells to become a particular type of subset. Because it's an intracellular microbe, the subset will become a Th1 cell, release interferon gamma, and in turn that interferon gamma is going to go back to help these cells that started the whole cycle in doing their job. So that's the overall what's called Th1 subset pathway. The important things to remember here, all in red, IL-12 is released. In presence of IL-12 is where the cells become get motivated to become the Th1 subset. Those cells go through the transcription factors TBET, STAT1 and STAT4, and these cells will then release interferon gamma, and that interferon gamma will provide positive feedback for the Th1 subset, negative feedback for Th2 and Th17, so other subsets.
Dr. Raje, just to double check, the IL-12, that's coming from the dendritic cells, macrophages, and natural killer cells?
Interferon gamma is coming from NK cells. So remember, the cytokines are not just from Th subsets. The ILCs also release some cytokines. So when there is a viral infection, for example, before the Th cells come into play, the innate immune system is going to be activated. At that time, the macrophages release IL-12, NK cells release interferon gamma, and all these cytokines start helping in fighting. Interferon will interfere with the viral replication, and so that's produced at the site of infection, but that also helps in providing that signal for the T cells to say, okay, it's interferon gamma that's released in that area. That means we need Th1 subset.
Okay, thank you.
Yeah, but IL-12, IL-18, interferon gamma comes from NK cells, but type 1 interferons can come from dendritic cells, which all of them play a role in that Th1 subset. The important ones to absolutely remember are IL-12, interferon gamma, but then as you see, IL-18 and type 1 interferons are also mentioned. So then say the Th1 subset is formed. What does it do? It helps in activation of macrophages to ingest and kill those microbes. And then macrophages release, that interferon gamma that helps with, the macrophages can come from two sources that I just talked about. So NK cells help in releasing interferon gamma, and then once Th subset is formed, that also helps in releasing interferon gamma. These cells can also help in activating the B cells, right? And then in that case, Th1 subset will help in class switching to IgG. Why is that? IgG helps in clearing some of these, making more antibodies. They will activate complement, and that complement will also act to opsonize. And say similarly, Fc gamma receptors will help in opsonizing the microbes so that they can interact with the Fc gamma receptors on the phagocytes and help in that phagocytosis. So interferon gamma can also act on T follicular helper cells as well. So the figure is just showing that APC interacting with naive cell, becoming a Th1 CD4 cell, then interacting with that APC because of that interferon gamma that's going to help in more activation of that phagocytosis. So once the function of the Th1 cell depends on these cytokines. Interferon gamma is the important one. The other ones that are can be released are TNF, that activates neutrophils and helps in inflammation, and then IL-10 provides the negative feedback. So it is inhibitory. And so we'll go ahead and talk about these cytokines. So we talked about some of the cytokines that were important for IL-2 proliferation and activation, and now we are talking about this interferon gamma that's important for the function of Th1 cells. So interferon gamma is a homodimer that's produced by these cells that we just talked about, and they interact with their receptors. The receptors have two chains, interferon gamma R1 and R2. So those two chains have to come together where the interferon gamma can attach to, and once it attaches to that receptor, like every other receptor we talked about, there is signaling downstream, and so there is dimerization of these receptors that occurs, and that activates these kinases JAK1 and JAK2 that helps in phosphorylation and dimerization of STAT1. STAT1 is our transcription factors that once it dimerizes, it goes to the nucleus and helps in transcription of genes that are important for the function of interferon gamma. We looked at this pathway last time, so I'm not going to go over that signaling pathway, but I want to focus on the functions of once that signaling goes through. The transcription of the genes that occurs helps in making the proteins that are important for activation of macrophages, activation of IgG class switching, and then helping in that positive loop, polarization towards Th1. So the proteins that are important, the transcription of proteins that occurs are proteins including some of the things that help with antigen presentation, such as TAP, proteasome, HLA-DM, MHC molecules, that second signal proteins such as B7, 1, B7, 2, and all of these proteins are going to help in those functions of interferon gamma. So the basic function of interferon gamma is class switching to IgG and activating macrophages. So now that the interferon gamma helped in activation of macrophages, what do macrophages do? So first, apart from interferon gamma, it's that second signal that's needed, CD40 ligand interaction, that helps in macrophage activation, and that also helps in some of the transcription factors that in the macrophages that will make proteins important for their function. So macrophages are known for inflammation, so there are going to be some inflammatory transcription factors such as NF-kappa B, AP1 that are going to be important. And what does macrophage need for its function? They need that machinery for phagocytosis, right? So they need phagocyte oxidase or FOX. And the other enzyme complex that's important for macrophages more so than neutrophils in their phagocytic function is inducible nitric oxide, so INOS synthase. Inducible nitric oxide synthase, or INOS. So INOX is an important enzyme complex apart from FOX system that these macrophages are going to use. They also have some lysosomal enzymes that are important. But that's the interaction. Those are the enzyme complexes that are needed. So the transcription of those from the downstream signaling from interferon gamma will help in making some of these proteins needed in these enzyme cascades. These enzyme cascades are going to be used in killing the microbes in phagolysosomes. And apart from killing the microbes, unfortunately, they can also cause damage to the normal tissues that we are very well aware of. So the figure here is just showing that information that I just talked about, but then there is this part B that talks about some of the roles in the cell-mediated immunity and how that occurs, some of the proteins that are made because of that signaling with interferon gamma. All right, moving on to Th2 subset. Any questions about Th1 subset? All right, we'll summarize this at the end of all three subsets, but we'll go back, go on and move on to Th2 subset. So as you guys mentioned, the microbes that are important to activate the Th2 subset are helminths, right? Some of the mucosal microbes can also trigger Th2 subset activation. And then we know that the allergic diseases are also somewhere where Th2 subset is going to be active. So the goal of these cells is to have activated eosinophils and mast cells so that they can perform their function. So that's the goal. Now what happens is, if there is, say, helminthic infection, that helminth is gonna interact in that interact with the innate immune cells. Also, there are some other sources from where some of these cytokines are released, such as IL-25, IL-33, TSLP. There is IL-4 that's released, and all of those cytokines, in presence of these cytokines, dendritic cells or other antigen-presenting cells will take that information. What kind of infection? Here it will be helminth. And where is the infection? With that, is it in the GI tract? Is it in the lungs? And with that information, it will go through the lymphatic vessels, get to the lymph nodes, where they'll interact with the specific naive T cells. Once they provide that information, these naive T cells, in presence of IL-4, will activate some of these transcription factors, such as STAT6 and GATA3. STAT6 and GATA3, these transcription factors will lead to transcription of certain genes that are important for the function of these Th2 cells. The most important part of that is releasing IL-4, and some of the other cytokines like IL-5 and IL-13. These Th2 cells, again, IL-4 will be the important one to help in amplification. So IL-4 is released from these cells and also acts on the receptors on the same cell to provide that positive feedback, so that the cell is committed to become Th2 cell, and will help in commitment of other cells, cells, to become Th2 cells, and they'll inhibit the Th1 and Th17 subsets at that time. So that's the goal of this pathway. Once that happens, the IL-4, IL-5, and IL-13 are the ones that are going to be important in the function, which means their function is to somehow go back to the cells that are needed, that where everything started, mast cells, eosinophils that are going to be activated so that then they can kill these helminths. So how do these Th2 cells function? We know that the helminths are too large. They can be resistant to the microbicidal activity of the phagocytes. So phagocytes may not be enough to kill these helminths, but they need these mast cells, they need eosinophils, and they also need IgE antibodies. So the function of these Th2 cells is to provide all those activities. So here are our naive T cells. They become activated, proliferate, and differentiate to become Th2 cells. Also, the follicular helper cells will, in that presence of those cytokines, will have the B cells class switch to IgE more than other subsets. So same thing for Th1, we saw they class switch to IgG. Here they're going to help in class switching to IgE. And then the Th2 cells will release that IL-4, 13, and IL-5. IL-5 will help in eosinophil activation. IL-4 and 13 will help in macrophages to have alternative macrophage activation, so helps in tissue repair. And then IL-4 and 13 will help in mucus secretion, right? Help in mucosal barrier function. That's going to be important for mucosal layers as well. And then IgE will help in mast cell degranulation. So all of that together will help in clearing the helminths. All right, so we're now going to talk about cytokines of Th2 subset. IL-4. This is a cytokine that's produced by Th2 cells, also comes from ILCs and mast cells, right? So more sources than one. And that cytokine is going to then interact with its receptor. The receptor has an alpha chain and a gamma chain. And then again signals through JAK1, JAK3, and STAT6. STAT6 will go to the nucleus. It's a transcription factor, helping the function of the cytokine. The IL-4 cytokine can also bind to IL-13 receptor. So IL-4 can bind to IL-4 receptor. It can also bind to IL-13 receptor. Function of the IL-4 cytokine: it helps in development and proliferation of Th2 cells. It helps in class switching to IgE, alternative activation of macrophages, helps in increased peristalsis of GI tract, and recruiting eosinophils. We know IL-5 is more important for eosinophils, and IL-13 is important for mucus production. So IL-13, again, structure very similar to IL-4, produced from various sources, and then it binds to IL-13 receptor, which has alpha chain of IL-4 receptor and IL-13 alpha-1 chain as well. Signaling through the same JAK-STAT pathway, JAK1 and JAK3 and STAT6, helps in making proteins, transcription of genes that make proteins that help in the function of IL-13. IL-13 helps in class switching to IgE and IgG4, alternative macrophage activation, helps in producing mucus, important in airways and GI tract, and then helps in recruiting eosinophils. IL-5, again, another cytokine produced by various sources, and then interacts with the IL-5 receptor. This has an alpha chain and a common beta chain. And then signaling goes through JAK2 and STAT3. And then function, we know it actually helps in class switching to IgA. That's important for mucosal immunity. Also helps in activating eosinophils, and important one for growth and differentiation of eosinophils. Eosinophils have Fc receptors for IgE and IgG, and so that they can bind to the opsonized helminths, the helminths that are opsonized by these antibodies. Then they come along and bind to the Fc receptors on the eosinophils, and then the eosinophils can release the chemicals needed. All right, we talked about and mentioned this alternative macrophage activation. So we know that apart from the inflammation that macrophages can cause, as we see in Th1 subset pathway, the macrophages, when activated by Th2 subset, instead of doing the same thing and help in phagocytosis, they help in collagen synthesis, fibrosis, and angiogenesis. So they help in that tissue repair, and they actually help, they cause release of inhibitory cytokines like IL-10 and TGF-beta. There are only two known inhibitory cytokines, IL-10 and TGF-beta. Those are the only two ones that we know are inhibitory in nature. Moving on to Th17 subset, same concept. We are going to start with a different stimulating signal. In this case, it's an extracellular bacteria or fungi, right? So say there is a fungal infection. In presence of that fungus, the innate immune system is going to be activated and release certain cytokines. Those are going to be IL-6, TGF-beta, two important ones. There's also going to be IL-1, IL-23, but the basic important cytokine milieu that's important for this Th17 subset is IL-6 and TGF-beta. In presence of that, the transcription factors that are going to be activated are Rho gamma T and STAT3. And these transcription factors are going to end up in transcribing genes that make proteins important for the function of Th17 cells. The most important of that being IL-17 and IL-22 cytokines. These will also provide that feedback positive amplification, as well as inhibit other Th subsets to be formed. Now, this is important in mucosal tissues such as GI tract, where there is high TGF-beta present all the time. In presence of just TGF-beta, remember TGF-beta is inhibitory cytokine, so it helps to maintain an anti-inflammatory environment. However, when IL-6 and TGF-beta come together at that time, there might be more Th17 subset that might be created. So let's look at the function of these cytokines and these cells. So the Th17 cells will recruit neutrophils and monocytes, and once they reach the site of infection, there are Th17 cells that will recruit these cells, but the phagocytosis also helps. So sometimes the Th1 subset also starts helping in killing some of these microbes. Th17 can also cause problems such as Th1 cells. If they are overactive, they can cause that granuloma formation from macrophages. Th17 cells can actually lead to can be involved in the pathogenesis of some of these disorders such as psoriasis or inflammatory bowel disease, rheumatoid arthritis, multiple sclerosis, apart from the beneficial effect of Th17 in presence of extracellular bacteria or fungi. Some of the other T cell subsets that we don't typically talk about are gamma delta T cells and invariant NKT cells, or sorry, NKT cells in general. So the gamma delta T cells don't have as much diversity as alpha beta T cells. When we talked about these CD4 cells and the Th subsets, we were talking about all of those cells that are single positive and they are alpha beta TCR positive cells. There are 5 to 10% of the cells that are gamma delta T cells. They don't have as much diversity. They don't have MHC restriction. They don't use MHC for their antigen presentation. They use something called non-classical MHC, and they can recognize things other than the peptides. Remember, the MHC molecules present antigens or peptides to the alpha beta TCR. However, the gamma delta T cells are not restricted to just recognizing peptides. They can recognize amines or lipids or phosphorylated molecules that can activate these gamma delta T cells. NKT cells, there are cells called invariant NKT cells that again do not have as much diversity. They do have the T cell, so they have the alpha chain associated with a beta chain, but they also have the NK cell surface markers. And they recognize lipids. They use CD1 non-classical MHC for presentation to them. And then there are some cells called MAIT cells. MAIT cells are mucosal associated innate T cells. I don't know what the I stands for. Sorry, I would have to take a look. I'm blanking on the exact name. These are actually alpha beta T cells. So the TCR is alpha beta, but they are still not as diverse. Clinical implications of some of these subsets. If the Th1 subset, so let's talk about X-linked hyper-IgM. So we know that Th1 subset interacts with the macrophages using CD40 ligand, and that helps in also helps in follicular helper cells, where they help the B cells in making IgM switching to other classes. If this is defective, there is no class switching, so there is high IgM and not enough G, A, or E, but also the function of macrophages is affected, because of which there is susceptibility to opportunistic infections such as PJP. That's X-linked hyper IgM. Then we also have other issues with Th1 subset. If it's not as effective, it may not clear mycobacteria as easily. So there is a disorder called Mendelian susceptibility to mycobacterial disease, and that can be caused by defects in IL-12 cytokine, interferon gamma cytokine, or its receptors. And there are several downstream molecules like STAT1 that can have some of these, loss of function issues, because of which they may not clear mycobacteria. Apart from mycobacteria, some of the downstream molecules, if affected, can also have susceptibility to certain specific microbes, such as Salmonella, or in case of IRF8, it can be viral infections, so on and so forth. So there are multiple genes in that pathway, in that Th1 pathway, that can lead to MSMD. Th2 subset, easy, you all know it can be involved with allergy issues. And then Th17, defect in STAT3, can lead to Job syndrome or hyper IgE syndrome. That's just a short list of some of the things that can happen. Let's go back to the questions. A major function of Th1 cells is to activate macrophages. Which molecules expressed by Th1 cells are essential for its function?
Is this D, interferon gamma, and CD40 ligand?
Yeah, D.
That is correct. Interferon gamma and CD40 ligand. Move on to question 2. Which of the following is one way a cytokine made by Th2 cells contributes to atopic disease?
B, IL-5 activates eosinophils.
That is correct. IL-5 activates eosinophils. IL-13 is important for some other functions, not nitric oxide. IL-4 induces B cells to IgE, not to IgA. All right.